Welcome. Thank you so much for tuning in. We're very pleased to have you join us for the Southeast Ocean and Coastal Acidification Network's State of the Science webinar series. The title of today's presentation is Oceans Acidic and Low in Oxygen, Lessons from Estuarine Organisms. This series is hosted by the Southeast Ocean and Coastal Acidification Network. In short, the series lays a foundation for the state of ocean acidification science in the Southeast region. These webinars are intended to create a dialogue among scientists to identify what is known, what isn't, and what research in other regions of the U.S. can be applied to better understand ocean acidification and its impacts in the Southeast U.S. With awareness of and access to the research and its applications and uses, webinar participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. I'm Jen Bennett-Mintz, Education and Outreach Coordinator for NOAA's Ocean Acidification Program, and will be facilitating today's session on behalf of the SOCAN Steering Committee. During the presentation, all audience members will be in listen-only mode. You are welcome to type questions related to technical issues or questions for our presenter into the questions box in the bottom of the control panel on the right-hand side of your screen. We'll be monitoring incoming questions and will respond to them or pose them to our speaker. We are also recording this session. The video recording and PDF of the presentation will be available on the SOCAN website. We are very excited to have Dr. Lou Burnett, Professor of Biology at the Grice Marine Laboratory, College of Charleston, and the Hollings Marine Laboratory. At the College of Charleston, Lou was Chair of the Department of Biology for 5 years and Director of the Grice Marine Lab for 20. Before moving to Charleston in 1991, he was on the faculty at the University of San Diego for 13 years. He received his BS degree at the College of William & Mary, his PhD at the University of South Carolina, and in postdoctoral research at the University of Aarhus in Denmark. He is currently the president of the Southern Association of Marine Laboratories and president-elect of the Society for Integrative and Comparative Biology. Lou has had a long-term interest in how animals adapt to estuarine environments, especially low oxygen and high carbon dioxide ones. So with that, I will let him share his interest. Lou, I'll go ahead and hand it over to you.
Okay. Doesn't seem to be responding.
Go ahead and perhaps just try clicking yep.
It worked before.
It sure did.
Okay.
Let's go ahead and maybe is anything working? Can you minimize the screen or minimize PowerPoint?
No. Yes.
Okay. All right, maybe from there, go ahead and try to move it into full slideshow mode and see what happens.
Okay.
There we go.
Right.
Perfect.
There we go. Well, good afternoon, everyone. It's a pleasure to be here. I've been listening in on the webinar so far, and I found them very informative and stimulating. I have worked since the mid to late 70s on issues related to hypoxia or low oxygen, and when I did my postdoc at the University of Aarhus in Denmark, I began working on acid-base physiology. So I was actually working on acidification issues long before it became popular. So I guess the lesson is, if you are patient, your research might become relevant. So today, I would like to go through a discussion initially of the co-occurrence of high carbon dioxide and low oxygen in coastal waters, especially because these two things often co-occur. And in the early years of my research, those of us who are working on low oxygen often considered low oxygen in isolation, and then later, starting in the 1980s, we became to understand that low oxygen and high carbon dioxide often co-occurred together, and I will talk about that. And then I will go through a couple of case studies, lesson 1, lesson 2, on oysters and crustaceans, highlighting some of the work that we've done in our lab as well as some of the work that others have done. Well, the most iconic symbols that we're all familiar with respect to issues in coastal areas are fish kills, and these fish kills are almost always associated with low oxygen events. And they're very iconic with the public. The public understands these images very easily. This is especially in the Gulf of Mexico, where there are these Jubilee events related to hypoxia, where there are large numbers of a variety of different kinds of organisms that are sort of jammed in together and ultimately succumb to low oxygen. And we know low oxygen is a serious issue in places like the Chesapeake Bay and here in this map in the Gulf of Mexico, where the issues have been extensively studied. In recent years, Diaz and Rosenberg have brought attention to the research community on worldwide oxygen levels and how the occurrence of low oxygen in coastal waters is on the increase, and this is not surprising, obviously associated with a variety of issues related to development. So low oxygen is an insidious killer, but to point out now that there are lots of sublethal effects on low oxygen, and those of us who have studied low oxygen have tried to investigate these. But these low oxygen events, at least in coastal waters, are nearly always accompanied by elevated carbon dioxide. And this pattern is a typical pattern. This is from Scott Lerberg's, one of our graduate students, from his work on Deal Creek in a tidal marsh, and you can see. Again, my laser here. You can see that there is a cyclic phenomenon to oxygen. So this is dissolved oxygen as percent air saturation. And I've given you here 3 levels. There's a normoxic level, something we call air saturation, or something that I consider moderate hypoxia, about 50% air saturation, and then severe hypoxia. And you can see that these periods of low oxygen correspond with nighttime. And the simplest and most obvious explanation for this is that at night photosynthesis is not occurring, and yet the oxygen demand for the organisms living in the water continues. In the daytime, when photosynthesis ramps up, it's fixing carbon dioxide and producing oxygen. And so this pattern is something that we see over and over again. In that same area, if you take the observations that people have made looking at oxygen of percent air saturation, and let me change it to, sorry, I'm going to get my colors sorted out here. If you change, if you look at oxygen in the water in terms of percent air saturation and simultaneous pH, you see that there's this very tight relationship such that at low oxygen, pH goes down, which is associated with the buildup of carbon dioxide. This is a very clear pattern showing that tight relationship between oxygen and carbon dioxide. So this is not only in coastal South Carolina waters. Recently, Frank Meltzner and his group published a very interesting study, and I like the title of this paper, Future Ocean Acidification Will Be Amplified by Hypoxia in Coastal Habitats. And I took one of his graphs and redrew it to show that same relationship with oxygen and pH. And to convince you that this is water that is not very warm, such as the water in coastal South Carolina, but this is water off the coast of Denmark and Germany, where temperatures are below 15 degrees, so this is occurring there. And so this pattern that we see of pH and oxygen persists even in European waters. Recently, I had the pleasure of participating in a NAML briefing on the Hill, and here's a link to these presentations. And Dan McCorkle of Woods Hole presented a graph that's very similar to the one that I just showed you from our data. You can see that he plots oxygen and pH, and the patterns, the day-night patterns persist. And if you look at that same relationship between oxygen and pH, it exists there as well. So let's move on to what we know about ocean acidification. And this is something that we've heard over and over again. These are numbers that are taken from the literature, showing what people believe to be in open ocean, the pattern that will occur. And I will use the term hypercapnia, so hypoxia or low oxygen or hypoxia, and hypercapnia refers to elevated CO2. In coastal South Carolina waters, then, you can see that this pattern already is much broader than the projected 2100 pattern, and hypercapnia in the coastal waters is pretty much a fact of life, and I've shown you some real data supporting that. So what I would like to do then is to talk with you about the response of organisms to habitat. So what do organisms do when the water CO2 becomes high and water oxygen becomes low? But there are also not only habitat issues, but there are issues associated with organismal life cycles. And I'll talk very briefly about that and spend most of my time talking about the habits of organisms. So my models for this will be the oysters and some studies that have been done on crabs and shrimp. So lesson one, onto the oysters. Some of you who live in this area may recognize this as a scene from Clam Bank at the University of South Carolina Field Station. And oysters there are intertidal. Oysters, approximately 95% of the oysters living along the coast of South Carolina are intertidal oysters. They're exposed to air twice a day. And that's certainly convenient for collecting oysters, but oysters are a convenient organism for going in, not only in the field, but taking them into lab. And you can do some interesting experiments on them. Here is a picture shown of me actually sampling hemolymph from an oyster in the field. But we've done some lab studies on this too, and I wanted to show you what happens when an oyster in the field is exposed to air. Remember, that happens twice a day. So if we take the hemolymph, or the blood of an oyster, and measure its pH, you can, and do this at different durations of air exposure, you can see that that before the oyster is air exposed, when it's submerged, it has a pH of around 7.3 or 7.4, which is absolutely typical of many marine organisms. But that when it is air exposed at 25 degrees, its hemolymph acidifies. Now this is a pH drop of 0.3 of a unit, And I'll remind you that pH is a log scale, so this is a large acidification of the hemolymph and the tissues that are bathed by the hemolymph. But look what happens if you increase the temperature to 35 degrees. Now these oysters are sitting out in the air, and when they are exposed to air in our hot summertime temperatures, their body temperatures can get well above 40 degrees. But at 35 degrees, look at the level of acidification. This goes down to well below 6.5. So this acidification is due almost entirely to the partial pressure of carbon dioxide, and that pressure of carbon dioxide is actually 20,000 microatmospheres, if you remember the table I showed you. So this is an extreme change that the inside of the oyster itself sees. If you look at the partial pressure of oxygen in those same hemolymph samples, again as a function of air exposure, you can see that the oxygen levels also go down in an oyster at both 25 and 35 degrees. And as it turns out, that during this 4-hour bout of air exposure, calcium ion concentrations in the hemolymph increase, and this is not surprising because calcium is being mobilized from the shell to at least partially help compensate for this acidification. So oysters see some pretty dramatic changes in CO2 that are not driven by the CO2 in the water in which they live, but by their habit. One of the effects of elevated CO2 and low oxygen on oysters has to do with their immune function. So what I thought I would do is to show you a graph of, this is called a ROI production, or reactive oxygen intermediates. It's essentially oxygen intermediates, free radicals produced by the hemocytes, or the blood cells of the oyster, that assist in allowing it to kill pathogens. And you can see that in a normoxic oyster, there's a certain value there, but when the oyster is exposed to hypoxia and hypercapnia simultaneously, there's a 33% decline in the production of these reactive oxygen intermediates. Now this kind of experiment has been done by a number of people, but we've done it in our lab on fish, on blood cells from fish, and we've also done it on shrimp and crabs. And the same pattern persists, that this production of reactive oxygen intermediates as a part of the innate immune response are very sensitive to both oxygen and hypercapnia. And one final bit of information on oysters: there's a disease called dermo, which is a single-celled pathogen, Perkinsus marinus. And I thought I would show you this study that a graduate student of mine did, where he was taking these Perkinsus cells and culturing them. And so if we count the density of the cells in the cell culture over hours of incubation, and he was doing this at 2 different treatments of carbon dioxide, 0% CO2 and 2% CO2, which is a high percent of CO2, but absolutely typical of what the oysters see. You can see from this graph that there's no real difference when the pH is held at 7.6. However, when we change the pH to a more acidic pH of 7.1 or even 6.3, you can see that there's a profound difference between the high CO2 treatment and the low CO2 treatment, in that the high CO2 treatment stops or prevents the cells, the dermo cells in culture, from growing. Well, this is actually very good for the oyster, because what it does is it holds in check the growth of the disease in oyster. And in fact, in intertidal oysters, at least in South Carolina, where I know the folks who have made the measurements, Perkinsus or dermo persists in nearly 100% of the oysters, but it does not reach very high levels. And we believe that this CO2 actually protects the dermo from taking hold. So the take-home lesson from the oysters: being hot and air exposed has some advantages for the oyster that I just told you about. It offers some measure of protection from the diseases, and some of the field values support this, although elevated CO2 and low oxygen appear to inhibit the innate immune response. Just a note on larval biology, especially in bivalves. A few weeks ago on this webinar series, George Walbusser presented an excellent summary of the state of what we know about bivalve larvae. And I wanted to touch upon that because I think there's another important point that can be made. So we know that bivalve larvae are sensitive to both hypercapnia and hypoxia. And what I thought I would do is to show you this graph from the study by Talmadge and Gobler in an excellent paper, in which they looked at hard clams, bay scallops, and eastern oysters, and they exposed them to ambient CO2, what they considered a mid-level of CO2, and then a high CO2. Notice that these CO2s are very low compared to some of the values that I showed you before. And if you could focus on the eastern oyster, although the others are interesting, you can see that over a period of days that the different classes of developmental stages of the oyster are shown to be really knocked back by the moderately high CO2 treatments. And there are a number of studies that have shown this, and George certainly outlined this, and actually gave some really interesting mechanisms to explain this occurrence. There's an older study that looked at hypoxia alone, and you can see that in this level of hypoxia, so this hypoxia was 1.5 milligrams, so that would fall into what I considered a severe hypoxia, although they did something called anoxia, which is very, very low oxygen. And you can see that this is percent settlement of oysters now. You can see that that in the severe hypoxia, that settlement was indeed inhibited, but that it was greatly inhibited with anoxia. So I've been perplexed by these kinds of findings until I thought about all the studies that I'm familiar with on larval survival and settlement. And pretty much nearly every kind of environmental variable you can throw at a larva results in some kind of diminution of survival. So different environmental variables have effects. So last week I was talking with Peter Kingsley Smith, who's at our South Carolina Department of Natural Resources, just across the street, and I was talking with him about this, and I was asking him about our oysters, Crassostrea virginica, to try to get a handle on, if things are so bad for the oysters, and the one I just showed you showed how bad things are for oysters at modestly high CO2 levels. If things are so bad for oysters, how do they survive at all? And so Peter and I had a good discussion on this, and he told me that oysters here begin to spawn in April and go through this process through August. So April through August, that's a large stretch of time, and during this time, and they produce millions and millions of larvae. So even during this time, if only a very small percent survive, which is probably the case, then they have succeeded. So even though the studies show that acidification has dramatic effects on larvae, so do lots of other things, and we just need to make sure we consider that in context. So I thought that was an interesting observation that Peter had. So let me move on to crustaceans. Our two crustaceans that we work with are the Pacific white-leg shrimp, which is the most commonly aquacultured shrimp in the world, and the Atlantic blue crab, which many of us living on this coast are familiar with. Both are good to eat, and our initial studies on the shrimp were one where we developed a bacterial challenge model since we were interested in the immune response. And what we did in the shrimp was to look at survival when we simply, over a period of a couple of days, when we just injected them with the saline and held them in under normoxic conditions and also in hypoxia at 2% CO2, and then injected them with this strain of bacteria, a Vibrio bacterium that is common, that is pathogenic to crustaceans. And you can see that injecting them with bacteria decreases survival, but that there was a significant decrease in survival when the shrimp were exposed to hypercapnic hypoxia. Same kind of thing was done in the very common grass shrimp, and you can see that injecting bacteria into them under hypercapnic hypoxic conditions has a profound effect. So shrimp and crabs actually have these blood cells or hemocytes, and there are typically 10 to 50 million per mil. And so this is a picture of some shrimp gills that I think is interesting, and it shows you the hemolymph channels that flow. These are finger-like filamentous gills, and you can see the blood cells over here. They're easier to see that are flowing into the gills. And so these are the blood cells that provide the immune response. In blue crabs, their gills have different shape. Instead of finger-like, they're leaf-like, so they have lamellar gills. This is a blue crab gill and a blow-up here. So that hemolymph then flows through an afferent vessel in a sheet-like fashion. And if you take a section through that, it looks like this, where there are these large hemolymph channels. And so here's a scanning EM of the gill for me to illustrate a point. Seawater's on the top side and the bottom side, and the hemolymph channel is in the middle. So we're kind of looking at this kind of picture here. And when you inject a crab with bacteria, just like we did with the shrimp, hemocytes aggregate around the bacteria and forming these hemocyte aggregations. These hemocyte aggregations are easy to visualize in the lamellar gill cells, and you can see one shown here. And eventually these aggregations form nodules, are absorbed into the epithelium, and ultimately molted away. But before that happens, the modules, as you can see shown here, were theorized to clog up the gills to prevent the gills from functioning properly. And we actually did a study on that. I'm not going to talk about the details, but what we found was that hemocytes do indeed clog the gills. We measured vascular resistance, and there was an increase in that, as you might guess. And there was a decrease in oxygenation at the gills, so the gills are not functioning in gas exchange, and a decrease in oxygen uptake. And another study done by one of our REU students a few years ago showed a decline in performance. So the immune response itself, which is good to get rid of pathogens such as Vibrio, but it has impacts on the organism. And I tossed out a number of references for you. All of these references are available at the end of the presentation. So we were interested in looking at performance in more detail. And a few years ago, one of my colleagues, David Skolnick, who is now at Pacific University, and in fact, he was one of my first undergraduates way back at the University of San Diego in the 1980s. David did a sabbatical here with me, and he was an expert on doing treadmill studies on a variety of organisms. So he built the shrimp on a treadmill, and what we have been able to do, mostly in blue crabs, as it turns out, is to do some interesting treadmill studies where we're able to look at environmental variables and the effects of environmental variables on performance. So here's an example of a blue crab showing you the effects of oxygen uptake, the ability to take up oxygen over time. During a 30-minute walking bout, the rate of walking in a blue crab, and I'll show you a treadmill shot in a minute, the rate of walking is 8 meters a minute, which is the equivalent of about 2.5 football field lengths in an hour. And you can see this is sort of a typical response you might find on any kind of organism doing exercise. And when these crabs are injected with Vibrio, they're still able to do the exercise, but their metabolism is impaired. So here's a shot of walking exercise in a blue crab, and the treadmill is sitting here. Water is flowing from left to right, and so I'll talk with you about what the blue crab has done. So this is a study done by Kristen Stover, one of my graduate students, and she published 2 papers in the Biological Bulletin, and what she did was to look at fatigue behaviors in blue crabs under a couple of environmental conditions. One of them was low oxygen, and she measured not only the duration of their walking, but how many fatigue behaviors they exhibited. And I won't show you a fatigue behavior, but a fatigue behavior for a blue crab would be reversing its leading and trailing legs or simply stopping. And what she found was not surprising, but here's a really interesting graph. So if you look at the time that the crab spent walking in 100% air-saturated water, you get, first of all, they can walk for up to 7 hours before they completely fatigue. If you lower the oxygen to 50% and then ultimately to 20%, you can see that they simply can't walk very long, 4 hours for 50% air saturation and only 2 hours for 20% air saturation. But what you also see is that these fatigue behaviors increase during this walking. Now, when we took this 50% air saturation group and redid the experiments but put them in hypercapnic water, water elevated with CO2, here are the results. The 50% air saturation water, the crabs were actually able to walk longer, and they had fewer fatigue behaviors. So this suggests that hypercapnia is good. So we were a bit perplexed by that at first, but then thinking about some of the other work that had been done on blue crabs, I was able to come up with an idea, and I'll tell you about that in a second. So the biggest thing, as I get toward wrapping this up, the biggest thing that we have found has to do with adaptations to low oxygen. Organisms respond to chronic low oxygen in similar ways. Humans do this too. How many of you have gone to places like Denver or Rocky Mountain National Park and tried to do some kind of activity? So in Denver, the mile-high city, the air is thinner there. If you go even higher in elevation, it's much thinner, and you can actually feel it. If you stay there for a period of days or weeks, certain things change in your physiology, and I've listed the things here. These are very common across many different kinds of animals. There's an increase in the respiratory pigment. So in humans, it's hemoglobin. For crustaceans, the shrimp and the crabs, it's another pigment called hemocyanin. Concentration of those pigments increase. The oxygen affinity, the attraction of the pigment for the oxygen, has an adaptive increase in affinity, and there's also an increase in vascularization of tissues. So in the blue crab, as it turns out, from a 1990 study by DeFura et al., they showed when the crabs were put in 35% air saturation, so something on the order of moderate hypoxia, that the crabs in fact increased their hemocyanin concentration from 3.11 to 4.40, and that was interesting. And there was this very adaptive increase in affinity. There were two kinds of increases in affinity. There was one due to lactate, urate, and calcium, so some specific things in their hemolymph. But the most interesting one for us was this increase in the structural change of the hemocyanin. So the crabs were actually changing their hemocyanin to make it a higher affinity hemocyanin. And just a quick reminder now, if you've taken a physiology course, you might be familiar with this. If you haven't, don't worry about it. I'll get through it quickly. P50 is a measure of the oxygen affinity. This is a relationship between partial pressure of oxygen and the percent saturation of a respiratory pigment. This is hemocyanin, but it could be hemoglobin. And the relationship occurs between the pH of the blood and the affinity of the pigment, such that there's this linear relationship. High affinity looks like this. Low affinity looks like this. The higher affinity is better for hypoxic response. And the point of this is that when we go back and look at the blue crab to explain why it can do better on a treadmill at 2% CO2, we see that there's an increase in oxygen affinity. This is from a study I did with Charlotte Mangum, my mentor and hero, back in 1986. And so going back and looking at those data, this explains why the crabs are better able to perform. But at the same time we were doing these experiments, we began doing some molecular biology here at the Hollings Marine Lab. There's a marvelous genomics core facility. One of our graduate students, along with a bunch of other people, did some early studies on going back to the Pacific white-legged shrimp. And what he found out was that hypoxia, which normally induces the production of hemocyanin, and it did, looking at the molecular responses, that the production of hemocyanin is actually muted by the presence of CO2. And this is a big deal. What we have since learned using high throughput RNA sequencing, and I really don't have time to talk about any of the data, is that indeed CO2 mutes the response, and we've done these, so you can see this little box here. We've exposed animals to 4 hours or 24 hours of either normoxia or hypoxia alone or hypercapnic hypoxia, and through a lot more molecular biology, we have confirmed that the results by Rathburn et al. Do indeed show that that the CO2 response, CO2 mutes the adaptive response of oxygen, of low oxygen, for many organisms, and that's a hypothesis worth testing. So, life in oxygen and CO2 environments that are more extreme and variable than the open ocean is tough. Immune functions can be compromised, and I've shown you that by hypoxia, and it's made worse by hypercapnia. And the immune response itself reduces performance, shown from the treadmill studies. And this performance is strained by hypoxia. Hemocyanin function, and this may be true of the hemoglobins, but it has not been investigated. Hemocyanin function may be limited by elevated CO2. And finally, the molecular approaches that we've recently embarked on will, I believe, provide many answers to these kinds of questions. I wanted to acknowledge our funding sources sustained over a couple of decades, And thanks very much for your attention.
Great. Thank you, Lou, for that very interesting and informative presentation, really getting at the relationship of oxygen, carbon dioxide, and some of the effects on organisms that inhabit our estuaries. We do have a couple of great questions in the queue. We are going to go ahead and open the floor for questions and discussion. So if you have any questions for Lou, please go ahead and type them into the questions box on the right-hand side of your control panel, and we'll go ahead and get those Tim. So to go ahead and open it up, Lou, Cheryl is curious about the C. virginica work and was wondering if you've run any metabolic assays. For example, what do you think the oysters are using to cope with, or are they using, I should say, metabolic depression to cope with hypercapnia?
Great question. Yes, they do. Bill Stickle and his group at LSU years ago showed using calorimeters, which is really the best way to get at metabolism, that because oysters are very good facultative anaerobes, they can get along without oxygen, and they use other mechanisms to do it. But one of the mechanisms that they use is they simply lower their overall metabolism. We've seen that in the crustaceans too, so definitely oysters are lowering their metabolism. But in another study that I did with Libby Wilson years ago, we looked at that specifically to see if there was a specific metabolic depression in oysters caused by CO2, and we didn't see any. Definitely they depress their metabolism with respect to oxygen, but CO2 doesn't appear to have a specific effect on metabolic depression.
Interesting. So Cheryl, if you have a follow-up question, please feel free to go ahead and type that in as well. And in the meantime, we'll move on to a question from Evan. In discussing how it's possible for oyster larvae to survive hypercapnic conditions, have you considered maternal effects? For example, mothers passing on along increased energy reserves to their young, genetic resistance, etc.
So I haven't done the larval work. I think it's very interesting, and that's a great question. People have considered maternal effects, and I think that I think George even addressed this in his webinar. And I know that that Donald Manahan at the University of Southern California, the other USC, has done some work on this, and I can't possibly summarize his findings, but I believe there are some well-documented maternal effects.
Great. So Lou, I just have it, and it's a sort of taking a step back. I don't know if this work has been done, but sort of looking at the diurnal cycles and the extreme levels of carbon dioxide, do you have any sort of insight or thoughts on what that will look like where that oceanic baseline begins to rise in that estuarine, sort of where the estuary meets the sea?
Yeah, I mean, the noise in what we see here is so large. I think the baseline has to increase as atmospheric CO2 changes. But remember that the gases in the atmosphere are rarely, if ever, in equilibrium with the water. But definitely they will have an effect. I'm not sure you're going to, and the effects we see may be on the larval end of things that appear to be exquisitely sensitive. So I see the baseline is creeping up, and I don't think it's going to really have too much of an effect on juvenile or adult organisms, but I think we will see effect on larval organisms.
Great, thanks. So if there are any remaining questions, please go ahead and type them in, and we will go ahead and get them to Lou. We'll just wait a moment or two to see if there are any remaining questions. All right, it looks like we have a, a quiet bunch this afternoon. Well, thank you very much again, Lou, for that really great presentation. If
You're welcome.
any questions do arise for in our audience after this webinar, please go ahead and feel free to email Lou. You can see his email address here, and he will, I'm sure, be happy to answer them.
Certainly.
And if you are interested in joining, I'll go ahead and shift gears. Yeah, we got a few comments, Lou, in the questions box. Great presentation. Thank you very much. So those are coming in. And for those of you who may be interested in joining the Southeast Ocean and Coastal Acidification Network listserv to learn about future updates of the network, please go ahead and sign up at the URL shown here. And you can also direct any questions about the network itself to the email address shown here, socan@secoora.org. We wanted to thank you again on behalf of the SOCAN Steering Committee for attending this State of the Science discussion today. We do welcome feedback, questions, suggestions for topics for the series, and you can submit input by simply replying to the follow-up email you'll receive in the next day or so. As I mentioned at the beginning of the presentation, the recording of this webinar and a PDF of the presentation will be available on the SOCAN website, which you can see here, the URL at the bottom of the slide. And we do have our next SOCAN State of the Science discussion in just 2 weeks from today. On Tuesday, June 16th, Scott Noakes of the University of Georgia will discuss an ocean acidification time series mooring at Gray's Reef National Marine Sanctuary. So down off the state of Georgia, and that will again be at 12 p.m. Eastern, and we really hope you will join us. Thanks again, Lou, for that great presentation. Thanks for the thoughtful questions and taking the time to join us. We do hope to see you at SOCAN's next State of the Science conversation, and we'll go ahead and wrap up the session now. Goodbye.
Goodbye.